early decades, and the things that greatly influenced pharmacology back then and still today is essentially the behavior of the subject. A lot of the technology now in my field, such as neuroscience, and I'm sure in your fields, are incredibly sophisticated. You can get down to very specific sections of mRNA that may ultimately change receptor expression that in turn you might think might change behavior. But the idea is, can you formulate some sort of therapy around such very specific and elegantly designed studies when it comes down to the human level? So it's no surprise probably that the pharmacology that changes behavior is probably the most influential still today. I don't think anybody in this room, even myself, I was born in 1978, really appreciates the revolution that happened in pharmacology around the 1950s. So some of the studies you'll see are from the 1950s, and it was happening at a time when there essentially was no treatment for neuropsychiatric disorders and other disorders out there, right? So, and it wasn't necessarily even considered a disease state. It was considered people who had these diseases were often shunned by society and essentially in some ways locked up into asylums. And it's very hard to find asylums nowadays. I remember as a kid, Dorothea Dix was still open in Raleigh, which was the asylum for North Carolina, state funded, and I think that's since been shut down. But essentially what happened through the behavioral pharmacology revolution and the idea that drugs can change people's behaviors essentially released hundreds of thousands of people across the country from being locked up into asylums to actually functioning out in society. And sort of that's what we were all born into. Even more sophisticated drugs now, but if these, a lot of people give pharmaceutical companies and other things a bad rap, and maybe it's deservedly so in some aspects, but without pharmacology, the world would look a lot different. And so we'll talk about some of those early things. So some of the stuff you'll see, in fact, all the behavior, all the data you'll see will be rooted in behavior. And it may look, to some of you, rather simple, maybe even rather boring, but this was revolutionary at the time. And it's quite interesting, and it was very important to understand what things affect behavior other than just the drugs that you give people. So some learning objectives for today is to describe the key principles of behavioral pharmacology. We'll do this at the beginning of the lecture, then we'll move on to describing general types of animal models of behavioral pharmacology. And then in a very, very general sense, and we'll spend most of our time here, and then we'll get a little more specific to describe animal models for antidepressant, anti-anxiety, and schizophrenic drugs. So there's two major objectives, or two ways to look at behavioral pharmacology. If you think of behavioral pharmacology like a coin, there's sort of two sides, both of which are encompassed by behavioral pharmacology. So behavior is used to answer a question where the primary interest is in pharmacology. Okay, in this case, you're not necessarily looking at behavioral outcome as like a disease state in this case. You're trying to understand how the drug works, right? So if you have some idea that, some hypotheses you didn't know at all that dopamine was involved in maybe some sort of idea of reward, right? So you may design something called a conditioned place preference study where you continuously pair some drug that influences the dopamine system with some part of a chamber, and you'll allow, and by associating it, you give the animal that drug and put them in that chamber repeatedly. And then when you put them on the other side of the chamber, you don't give them anything. And then you later in a test, you ask the animal where it wants to go, and it goes and hangs out where you paired the drug, the context in which you paired the drug. And that drug, hit the dopamine system, for example. You might suspect then that dopamine is involved in something that the animal wants to be around, right? In the very crude sense possible. You might do other behavioral tasks to sort of narrow down that mechanism of what dopamine is doing. But that's behavioral pharmacology. In that case, you're just manipulating the behavior to understand the drug itself. The other way is more the clinical approach. So the behavior of the individual, the human or the animal, whatever, is the primary interest. So, and the drugs are used to dissect and elucidate certain behavioral phenomenon. So really what you're interested in is actually manipulating the behavior with the drug, okay, as opposed to understanding the drug with behavior. Sort of two sides of the same coin, and probably a lot of overlap and relatedness there. So some important principles of behavior is that the idea for behavioral pharmacology and research in this area is that it should be objective, okay? Quantitative, what you really want to do is sort of get some numbers out at the end. You don't really want to give qualitative reports, although those are helpful in the end, you really need to be able to show a shift in some metric that is observable. Stable over time, so when you're trying to understand shifts in behavior, you really need to explain behavior out of the context of the drug and then be able to apply the drug and see if that behavior shifts. So you need some stability. You can't just have, for example, a baseline of behavior where somebody shows depression one day and no depression the other day, and mild depression the other day and then schizophrenia the next day. In a very crude, rough example, right? You can't really test any sort of pharmacology or behavioral pharmacology under those conditions. Reproducible. Okay, that's some of the principles of behavioral pharmacology, the same as you'd see in other science. You can manipulate these behaviors over a range of parameters. We'll get into that a little bit later on. So you can manipulate multiple different parameters to change behavior, not just one. And sensitive to a number of pharmacological and environmental interventions. Now, drugs can act, not only do drugs influence the system, in the sense that they make you feel different or change behavior, but they can actually be part of the environment as well, right? Particularly with drugs of abuse, they can act as stimuli. They can cause things in people when drugs are paired with certain environments that will elicit certain behaviors in one context, but won't elicit behaviors in another context, okay? So we talk about these as unconditioned stimulus effects. Unconditioned, this is going to be a word. Conditioned and unconditioned are going to be two words that are used a lot today. Unconditioned means essentially that it's, I would think of it, and the way I would describe it, as sort of a natural response, something that hasn't had to be learned by the individual. It's sort of innate to the person. They say that drugs can elicit responses. Usually the word elicit is associated with unconditioned stimuli. If something is said, an unconditioned stimuli is said to elicit an unconditioned response. There was no learning, nothing going on here other than the natural innate response. You can have reinforcing stimulus effects. So, for example, the drug itself plus the presence of it, or the removal of a drug in the context of someone who's had a drug on board, can ultimately be a reinforcer. A reinforcer, we'll talk about in a minute, is anything that increases the probability of behavior happening again. So it's reinforcing if the drug or the presence of the drug or the absence of the drug increases the behavior, some subsequent behavior, okay? It also has discriminative stimulus effects. What does that mean? Well, drugs can set the occasion for other behaviors, okay? So if you have somebody, let's say, who's a drug addict who takes cocaine, they may act very differently when on cocaine than when not on cocaine, but some of those behaviors in which the person may act in the context of being on cocaine aren't exactly a direct result of the pharmacology of cocaine itself. They may have come to learn something in the context of being high on cocaine that will increase the probability of those responding happening again. Let's say crime. Cocaine as a pharmacological agent doesn't necessarily increase crime in a pharmacological sense, right? However, people who get together and maybe do drug X a lot, they may learn in that context that crime, I don't really want to attribute any psychology to it, attributing what their thought process is, but it just in general increases the probability that crime would happen. But it's not necessarily the pharmacology of cocaine to do such a thing. Okay, it may be a learned behavior that these things are associated with being high, but then when somebody is not high, it may be not criminally motivated at all and have no intention. So learning, it's almost like being a different person in a way, being under drugs versus not, right? So, two things you'll have to know for the test. You'll have to know a little bit more for this test than the other one, but I would start focusing on these slides forward, and I'll tell you when to stop. But we really need to understand, and the whole point, and I think everybody should really understand, no matter what your profession is. If you're going to be a counselor, you need to understand these sorts of mechanisms. It forms behavioral therapy, it forms pharmacology of all kinds of things. If you're going to be a medical doctor, you need to understand the behavior of your patients and things that influence these behaviors. So one primary principle, one of the earliest understood principles of learning is called classical conditioning. It started with the stimulus continuity principle, okay? So basically contiguous stimuli become associated no matter what you do about it, okay? If you repeatedly pair two stimuli in the environment over and over again, okay, those ultimately will become associated, okay? This is not necessarily conscious action. You don't decide that the two things that are paired will, become associated with each other. lot of examples of this in experiments. So this happens across all species, humans, mice, all the way up to humans, actually even non-vertebrates and everything. So we have what's called a neutral stimulus. If anybody has any questions, by the way, raise your hand and have me explain some details we go on. So a neutral stimulus is a stimulus that doesn't necessarily mean anything to the organism. So let's take, because we're going to get into some Pavlov's work, let's talk about Pavlovian conditioning of the dog, right? Pavlov's dog. So essentially we have a bell right here, and a bell to a dog really has no inherent meaning, other than all organisms, including the dog, will give something called an orienting response, or an OR. So when it has no inherent meaning or anything like that, we essentially call it a neutral stimulus. So it has no valence, it's not positive, negative, it's just sort of a neutral stimulus in the environment. And what happens in the organism is where it exists. I mean, it has sensory, so it knows a bell exists. It may have an orienting response, meaning it will turn and look at whatever noise is coming from the corner of the rib, right? Then, independent of that, no bell here, you have something called, again, the unconditional reflex. I gave the knee example here, but in the case of the dog with the famous Pavlovian conditioning, you have food. And the dog cannot control it, it's not conscious, but the dog will salivate when presented with food. So it's an unconditioned response, meaning, or you are. So the response didn't require any learning, it's innate in the animal, and it facilitates feeding for the animal, okay? The food in itself is considered in the context of salivation, okay, in an unlearned environment, an unconditional stimulus, okay? Yeah.
So with this example you just gave in your previous one about unconditional reflexes, both of those, at least as far as I know, are pretty physical neuronal pathways. Is that
right.
Always true?
Yeah, well, I mean, it might not be neuronal in the sense of central nervous system. So, for example, I'm not sure. In fact, I'm pretty sure that, for example, the kick reflex only goes to the level of the spinal cord, so it's more peripheral nervous system type stuff.
Okay.
But and there's some reflexes, for example, they call your gut the second brain. There's certain areas of your body that will control reflexes outside of the brain, so the brain doesn't have to deal with it even unconsciously. But yeah, there is some neural-like, whether it be peripheral or central system, causing these reflexes.
It wouldn't be something like what was the word I'm looking for? I'll think of it later.
Okay.
Carry on.
So, this can confuse some people, particularly if you've never had this before, because you'll see the conditional stimulus and the unconditional stimulus will ultimately be the same, can be the same, very similar things in the environment. It just sort of depends on the response, my apologies. The unconditional and the conditional response is going to be the same thing eventually. It just depends on what is eliciting the response. With the unconditional stimulus, such as food elicits the response, is considered the unconditional response. There's no learning here at all. Food naturally creates the unconditional response of salivation. Now during conditioning, remember the SS contiguity principle, stimuli are associated in time or paired. Here in conditioning, classical conditioning, okay, this is all classical conditioning for your test. The bell immediately precedes the food and is over many trials is paired with the food. So a bell happens, then the animal is given food. A bell occurs, the animal is given food. A bell occurs. What was once a neutral stimulus in the first, so this is the first trial, the neutral stimulus, then is followed by the unconditional stimulus here, and then is given an unconditional response. That's the first presentation, right? Then over multiple presentations, what you get is a conditional reflex, okay? Now, not only does the food promote salivation still here, but the bell will actually cause the animal to salivate, okay? So now we get the idea of a conditional response, a CR. So salivation is both a conditional response and an unconditional response, depending on what it is that elicited it. If it had to be learned or it had to be conditioned, it's a conditional response. If it's unlearned or it's unconditioned, it's an unconditional response. And if you look at part B down here, you don't even need the food present. It's not as if food has to be present in the trial. You can bring the animal in, have no food in the entire building, and a bell will cause the animal to salivate, okay? So this is classic where the CS, conditioned stimulus, causes a conditioned response independent of any sort of unconditioned So this is Pavlov's original data, right? So he had 2 dogs, dog 1, dog 2. On this axis, what we see is the number of drops of saliva across 30-second period, okay? Well, in a total of 30 seconds. And then on the bottom axis, you see essentially the number of conditioned stimulus, unconditioned stimulus pairings. And what you get on when is 0, no trials, essentially the animal's not going to salivate to a bell, right? So you're at 0, makes perfect sense. But 10 pairings, only 10 pairings already. Animal 1 is up to 20 saliva drops to a bell. 1's up to 10. 40 pairings. I'm sorry, this isn't 30 seconds. Yeah, 10 pairings, 20 pairings, 30 pairings. Speech maximum. All it takes is 30 pairings in the animal. So this is a very rapid process. Okay? This isn't something that you have to spend days and days and days with an animal. You can do 30 pairings in a day. And then you see the latency of the conditioned response, or the latency of essentially the drop of saliva to the bell. Takes, well, there's nothing in the first one, obviously, but after 10 pairings, you see it happens after 18 seconds, and then you see it gets faster and faster to the point within 2 seconds of the bell, you'll get the salivation from the animal. So this is learning. This is one of the first documentations in a laboratory of a learning process happening by revolution. There's aspects, you don't necessarily need to know this for the test per se, but I think it's really interesting to understand there's compounding that's associated with learning. And so people, obviously from here, a new science was born, and people are trying to understand under what conditions learning would happen, right? And you can start manipulating some of these things in very basic ways. They're actually quite interesting and stood the test of time and still exist in the labs today. So you have something called overshadowing, right? Where you only actually need one predictor, and only one predictor will happen with respect to the unconditioned stimulus. So when I first say shock, it's really shock avoidance. Animals, well, actually in this case, it is the presence of a shock that exists. So you can actually, animals can learn about aversive things too, and then take action to avoid those aversive things. We'll talk about that in a little bit. But so what we have in these experiments is a tone and a light that comes on simultaneously for the exact same amount of time, and then a shock will happen. Well, what will happen is the tone will ultimately elicit the avoidance response to avoid the shock. So there's learning here to the tone, but the light, when presented individually, you'll get no conditioned response, and the animal will ultimately be shocked. Because, then we'll say here that the tone overshadowed the light in learning. The tone was sufficient. So, for example, in the real world environment, if you go out and see people in the city carrying a lot of umbrellas and also a lot of dark clouds repeatedly, and those were always paired together, then you would only need one of those sufficient to tell you it's going to rain. Okay? And one of those would be overshadowed if it was always paired together. Okay? So you might learn that it's going to rain from dark storm clouds, but if it was always paired together, you would be blinded to the fact about the umbrellas. Okay, so umbrellas in the context alone would be predictive of the rain in that particular case. Now, it's not the way we really learn in the real world environment. Those things are also independently predict rain. So that's the difference than being concurrently on. There's something called blocking, where if you have a light, again, light didn't show anything here, but when light is given alone, it will predict shock, right? So the light will predict shock, so the animal will avoid the shock when the light is present. It will learn that. When then you give tone, now you give the tone at the same time as the learned light, ultimately the tone will have no conditioned response. Okay, so this is called blocking. We're saying that the stimuli 1 blocked learning of stimuli 2 because you had already learned stimuli 1 beforehand. So it's just a matter of when you presented it, whether the animal is presented at the same time, which one does he learn, or you have the animal learn something and you block something else from being learned. There are types of conditioning. Again, a whole field was born out of this, and people manipulate all kinds of things. Configurations, when it's presented, etc. Simultaneous conditioning, in this particular case, so this is what we were just showing before, the bell would perceive the food, immediately perceive the food, and they're saying simultaneous conditioning, that type of conditioning happens within 5 seconds. Typically what it is once the bell goes, it's like almost immediate, the bell and the food, or it's at the same time. So the bell and the food happen together. This is not the best type of conditioning. If you wanted to teach your dog like little tricks and things like that, this is not simultaneous presentation. Is not necessarily the best. They will engender learning, but it takes a little longer. Trace conditioning is essentially putting a time lag between the conditioned stimulus and the unconditioned stimulus. Delay conditioning is the best, the strongest, and the fastest type of conditioning, where you have essentially the conditioned stimulus come on for some period of time, it's extended, and then immediately switches to the unconditioned stimulus presentation with no time lag. This is very strong, and very strong learning happening here. Another type of interesting learning that happens with respect to this is that the animals will, or humans will learn to inhibit responding until the conditioned stimulus terminates. Okay, so they won't necessarily, if a lever is present, pressed in order to receive food, all through this whole time, they won't necessarily press a lever over and over and over and over and over again. They'll wait. Okay, so they understand the concept of time, waiting for it to go off, and that's the trigger for them to hit. Okay. Temporal conditioning, the unconditioned stimulus can ultimately become a conditioned stimulus itself. So this is an idea that animals and organisms understand the concept of time to some degree. Okay, so if you have a constant interfood interval, this constant is important. So every 3 minutes, you're giving a food pellet. The act of giving a food pellet can predict the subsequent food pellet that's coming up. Okay, so it can an unconditioned stimulus can be a conditioned stimulus itself. Backwards conditioning. I don't think I've read a paper on conditioned stimuli, backwards conditioning. But essentially the idea is the unconditioned stimulus comes first. Again, it's close enough in time for the contiguity principle to happen where these things become associated. And differential conditioning. Okay, so you have two conditioned stimulus. One conditioned stimulus will predict food here under a simultaneous conditioning parameter here. And then you have a second conditioning that routinely does not predict food. Okay, so then you get situations, which is really nice in experiments, where you have a positive conditioned stimulus and a negative conditioned stimulus. You can show that the animal, you can predict the availability of something, and then you can predict the lack of availability of something with a different tone. Yes?
For temporal conditioning, does it matter how long the interval is between?
That's a good question. I would imagine yes to some degree. It can't be, we'll talk about intervals in a little bit with schedules of reinforcement, which might end up answering your question, but it has to really, animals in particular, it has to be predictable in order for the unconditioned stimulus to sort of act like a predictor. And then, if you're talking days here, I don't think the animals, there has to be some sort of contiguity there. So I don't exactly know what that threshold is, and it wouldn't surprise me if someone's discovered that, and I don't know. So the answer to your question is yes. I just don't know exactly where it is. So this is all classical conditioning, right? This is associating things in your environment with other things, and then formally neutral things in your environment take on meaning. Now we're going to talk about operant conditioning, which is a different type of learning paradigm. Okay, it's also called instrumental conditioning. Okay, it's based on the principle that behavioral is the result of its consequences. Okay, so some behavior happens, and there's a consequence to that behavior, and the subsequent behavior is either going to happen or not happen, again, because of the consequence that happened to that initial behavior. Okay, so there's 4 different things to know here. I think putting it in this table is the easiest way to demonstrate it. Okay, so you have something called reinforcement. Reinforcement is the idea that the behavior is any time that the behavior, you increase the probability that the behavior is going to happen again in the future, which is indicated by the up arrows. So the arrows indicate whether behavior is increasing or decreasing. So under the reinforcement column, you can see in both cases the behavior is increasing. Punishment is sort of the opposite of reinforcement in that matter. Punishment is going to cause the behavior to be less likely to happen in the future. It's not saying it's not going to happen, you're not going to eliminate it necessarily. It's just going to decrease, you're talking about probabilities here, you're going to decrease the probability the behavior is not going to happen again. Okay? So you see the down arrows in that column, right? So we distinguish reinforcement from punishment. They're sort of opposites of each other. Now this can get kind of confusing if you've never seen it before, so make sure you pay attention. Then you have the idea of the words positive and negative. Positive and negative, and this does not refer to whether the behavior exists or not. Don't get confused. negative reinforcement and negative punishment, okay? So positive reinforcement is presenting something in the environment that didn't exist to increase the probability that a behavior is going to happen again. That's positive reinforcement. You can get that definition just by looking at sort of the table here, the graphical form here. So giving a kid a piece of candy, the presentation of something that wasn't there, the candy, okay, to increase the chance that when they said something nice, they'll be nice again in the future, right? That's positively reinforcing that individual. Removal of something to increase the behavior, for example, this is sort of removing, so in drug addiction, negative reinforcement and positive reinforcement is big as well, but negative reinforcement is a huge model that accounts for like at least 50% of the behavior out there. That suggests that people will self-medicate. So they feel really bad about themselves, and so that bad feeling increases the probability that they will take more drug because they want to alleviate negative symptoms, right? So the removal of something increases the probability that behavior will happen again. They took a drug, they feel better, okay? They've removed the negative affect associated with their current state, and removal of that is going to subsequently increase the probability that they're going to take that drug again in the future. Okay, does that make sense? Okay, another example. So you want, well, gotta be punishment, let me think.
The definition is taking something away to enforce a behavior?
Yes, essentially.
Okay.
Yeah.
Does it always have to be something bad? Always taking away something that's negative?
No, I don't think it always has to be bad.
Is
I feel like that's a relative term.
I would
Well, that's
imagine
a good
that's the
point.
easiest way.
what if like you take somebody's license away to enforce them? I don't know, I'm trying to think about it in like regular terms.
Yeah, okay, well, let me talk about all four.
Okay.
So you can have a context for which all things. So,
Okay.
presentation of something, positive punishment, that's what most people think of as just simply punishment. It's really called positive punishment, but it's, you don't, you want to, your child, hits somebody on the playground and you spank them or you do something like that. The spanking is the presence of something that wasn't there before, and you're trying to reduce them from hitting somebody else, right? So you're trying to decrease the behavior by punishing them. Incarceration, right? Jail. You're putting somebody in jail to decrease the probability of subsequent behavior, presumably, in a therapeutic approach as opposed to just keeping them away from society, right? Negative punishment is the removal of something to decrease. So your child hits somebody, you want to decrease that behavior, so they used to have the bite all the time, and you take their bike away from them as a punishment. That's negative punishment. You're removing something from the environment to decrease subsequent behavior. Okay? Yeah.
Is there any evidence to suggest like which one of these works best for people? Do people mostly respond to one or the other? Or?
Yeah. So, well, it's been known for a long time, for example, let me answer that in the opposite way. Punishment does not work long term. Okay? Usually what we know with the research now is it happens in the context of an environment in which the person feels like they're going to be punished, but they don't take it outside of that context. Does that make sense? So it's very context specific. So we've known, for example, for decades, independent of the politics of all that, I'm not talking about policy here and how you want to go, but for example, jail time does not work for drug offenders at all. The recidivism is extremely high, and like 80–90% of people who are in jail for drugs will go back doing drugs when they're done. Okay? So that was a positive punishment. A real world example of positive punishment is simply just when they're out of the now they won't do drugs necessarily in front of a police officer or in the context of the jail, right? Because that's very context specific, but it's hard to make that manifest when they're independent of that, right? Positive reinforcement is huge. So you have token societies, especially for children, to learn how to do better in grades. They can earn positive, it's highly influential, positive reinforcement. And it's really used in a lot of behavioral therapies today. So let's think of do we still need to talk about negative reinforcement?
That's okay.
Okay. I didn't know if it clicked from learning all the different ones or. Okay, so basically opera conditioning, anything we're talking about is adaptable to essentially all species. It says so. patterns of responding can be varied, and we'll talk about that in a little bit. And then you can get stable periods of responding and reinforcement over long periods of time. You can have it within sessions, and you can even take it over weeks, days, months, and even years. Okay. So within sort of the idea of instrumental conditioning or operant conditioning, we have things that are often used, and this is sort of models of drug addiction. It's not specific to drug addiction necessarily, but when you want to model drug addiction, you want to come up with treatments for drug addiction, you want to understand drug addiction, a lot of these schedules are used. So schedules of reinforcement. Okay, so now we're only talking about the reinforcement side, the idea that you are increasing the probability of behavior. And there are certain schedules. Schedules are simply just ways in which things are presented, or the stimuli are presented, that engender different types of reinforcement. Know these first four are the most important, in my opinion, for you to understand. These are sort of nuanced, and if you're in the field of drug addiction, you may want to know these, or if you're interested in learning, you may want to know these. But I think for the purposes of the general field here, everybody's varied interest, these things, the first four are the most important. So we'll spend more time here on these first four. So these are classic responses that happen under these first four. We're going to talk about schedules of reinforcement. Essentially what you have is the combination of two different words, fixed and variable, ratio versus interval, right? So you have fixed, which means either the amount of responses that's required to receive a reward or a reinforcer, like food, for example. The amount of times you have to press the lever is fixed over time, okay? Variable means it's not fixed. So the amount of responding that one would have to make on a lever, an animal would have to make a lever, changes across the session. So in the fixed interval, what people refer to as like fixed interval 1, for example. Fixed interval 1, the animal would have to press the lever one time to receive a reward, or like a food pellet. Then you would eat the food pellet and then press the lever one time, get another food pellet. That's a fixed ratio 1. Fixed ratio 5, they'd have to press the lever 5 times. So the ratio is always the same, it's fixed, right? A variable ratio is those requirements, those response requirements shift across the session. So the first trial in the session may be a fixed ratio 1, then it'll be a fixed ratio 20 once the animal eats the food pellet. Then it'll drop back to a fixed ratio 5. It can vary across time. A fixed interval is regardless of how much the animal responds on a lever, okay, or a keypad or anything like that, it's still, there has to be a certain amount of time that passes that's always the same in order to receive the reinforcer, regardless of how much the animal responds, okay? So you could do a 3-minute fixed interval. So it doesn't matter how much the animal presses in 3 minutes, the animal's not going to get a reinforcer until the 3 minutes is up, right? Variable interval is sort of like fixed interval. There's an amount of time between, but that amount of time is variable. For the first trial, it may have to wait 3 minutes. For the next trial, it may have to wait 20 minutes. For the next trial, it could wait 30 seconds. So that's variable, okay? Now, these are the types of responding you get on a lever for an animal when you engender these sorts of schedules, okay? So on a fixed ratio, now these are responses, 100 responses on a lever, let's say 200 responses on a lever, and this is time across this axis. So every time it sort of inks up, you sort of get these are more and more responses, right? And the little dash right here that's sort of diagonally to the right, those are when it receives a reinforcer, okay? So what you typically get under fixed ratio schedules here, I don't know what it is, but I guess this is 50 responses, so it looks like this ratio would be fixed to 25. This looks like a fixed ratio 25 because the reinforcer is fixed and it's sort of given every 25, 50, 75, 100, right? So you get this continuous sort of responding on a lever. The animal just keeps sort of pressing, sort of continuous over time. But what you get sort of that's unique to the fixed ratio is this post-reinforcement pause that a lot of people have tried to investigate what the heck is going on with this. So once the animal receives the reinforcer, like sucrose pellet or fruit juice coming down, why do you have this brief momentary pause? Is it satiety from receiving the reinforcer? Is it exhaustion from the animal is taking a break and it needs to, these sorts of things are, and it turns out, which is really interesting, that the further away you are in sort of some element of time or in terms of number of presses, the less reinforcing the reinforcer is. So they really narrowed it down to the idea that this is, you don't have to know this for the test, but I find it really fascinating that this sort of lack of responding is sort of a lack of motivation to ultimately receive the then as it presses, it realizes it has one less press to do. But then by the time you get to, 20, 19, 18, it'll start pressing more at a continuous rate because it's getting closer and closer. And the proximity of the reinforcer to where the animal is in relation to its responses dictates the amount of motivation the animal has. So when you start manipulating these sorts of curves of drugs, you might be able to start manipulating motivation and things like that. Okay? This is how we sort of you can manipulate these aspects of the reinforcement part of the curve in order to get at mechanisms for what's happening. Yes.
So ratio is response dependent and the interval is response independent?
Yes, exactly. Interval is time dependent. And variable ratio, okay, this is actually probably what happens most of the time in the real world. Gambling is a good example of this. You don't know when you are going to be reinforced. But you're going to only be reinforced after you make some sort of response. Okay, it's response dependent. You can't just sit there and receive money usually. Also drug addiction. Usually responses some effort has to be made to get drugs, but you're not always doing the exact same thing every single time in a drug context and always getting the drug, right? I mean, cocaine may or may not be available depending on dealers and things like this. This is incredibly strong. Look at the responses here. Incredibly strong responses. No post-reinforcement, pause, and just constantly pressing on that lever. Okay? So when the animal cannot predict when it will receive some reinforcer based on its responses, it will just sit there all day and press the lever continuously. So then we have fixed interval. Okay, so these are time scales. You get consistent responding under interval scale, interval schedules. When it's fixed, in other words, remember how I told you earlier, animals are pretty good at predicting how much time has elapsed, which is really, I think, elegantly shown here into the fixed interval. So I don't know what this is, 180 seconds, I don't know, whatever. Fixed interval, 90 second, maybe type thing. The animal essentially doesn't respond after receiving a reinforcer. It has what's called scallop responding, scallop here, some sort of U-shaped thing. No responding, and then as the time approaches, the animal's conserving its energy. Essentially, it's not going to start pressing the lever until it knows around the time in which it's going to be able to receive that reinforcement. All the animal has to do is press the lever one time, but it has to wait 90 seconds, but it doesn't know exactly 90 seconds in its head. So it's kind of like when you first start class, you don't really look at the clock, but once you start getting bored with me and then an hour and a half has gone by, you may check the clock like 6 or 7 times, every 5 minutes. And so your responding is going up and up and up as you get closer to the reinforcer. Variable interval, essentially the time changes in an unpredictable manner. You get this nice continuous responding of the animal, but certainly not as vigorous as when its responses actually matter, right? I mean, its responses do matter, but it knows that it can't predict it. The variable interval and variable ratio are hard to train, so acquisition is slower than the fixed, which might make some sense, right? But it's very hard to extinguish. Extinguishing means if you take the conditions stimulus, you take the, if you stop pairing these sorts of things like the lever response and cues that will predict the availability of reinforcer, you take all that away and just present it on its own, the animal will essentially no longer associate the two. Over some long, that's called extinction, okay? And it won't necessarily respond on a lever if the lever's not giving it reinforcers, right? Variable ratio and variable interval are very hard to extinguish behavior. They're very resilient, resistant against extinction. You'll just keep pressing a lever. But they're hard to train the animal on. It's a slower acquisition. These are easy, but if it's on a fixed ratio 5, and then all of a sudden it doesn't get any more after a fixed ratio 5, it'll just stop. So it's not as resistant to extinction. Again, this is just a distribution of interresponse trials on a DRL schedule. A DRL schedule, I don't know if I really want to talk about that too much. Essentially, it's sort of in some way, it's a punisher. So the animal has to wait some period of time, let's say 30 seconds, and it can't respond at all. So it has to inhibit its responding in order to receive a reinforcer. So it has to only respond after 30 seconds. And so if you have a DRL 20 second, you can see that the animal can tell most of its responding occurs in this histogram right around 20 seconds. Progressive ratio schedule, you don't, this is when the reinforcement is contingent upon increasing number of responses. So the first lever press, the animal will receive a drug like cocaine or receive some sort of reinforcer food. Then on the next trial, the animal will have to press the lever 2 times. Then on the next trial, have to press 4 times, then 8 times, then 16 times. So you get increasing amounts of the ratio that it would have to, and then you can see how much the animal will work for a particular reinforcement. It's usually an indication of motivation of the animal for a particular reinforcer. An animal will work more for cocaine, so they can get ratios as high as 1,500. So the animal will have to press the lever about 1,500 times to get 1 infusion of cocaine. You do that with food, and an animal that's full is not going to press the lever 1,500 times. So in that sense, you can test the degree to the reinforcing efficacy, we call it. How reinforcing is a reinforcer, right? You can start to compare these things. Concurrent schedule, where there's 2 or more reinforcers available simultaneously on different operanda. Operanda are just basically the things it uses in the environment to get the reinforcement. So 2 levers would be available. 1 lever will give you cocaine, 1 lever would give you food. Which one does the animal choose? Which one does, under what conditions can you switch the animal from food to, to cocaine? So in choice trials, what we do in drug addiction, for example, is we want to make cocaine not interesting to an animal who's had a lot of cocaine. So we give it food on 1 lever and we give it cocaine on another, and then you inject it with drugs, and the animal all of a sudden switches from all this interest to cocaine and it'll start to take some food instead. So you're sort of devaluing cocaine in the context of certain therapies or certain drugs, right? So that's where a concurrent schedule would be helpful. There's multiple, there's something called multiple schedules. So multiple schedules is sort of different than concurrent schedules. Those concurrent schedules can have 2 things going on at the same time, right? An FR5 on one and an FR5 on the other. Multiple schedules is where 1 operanda or 1 lever will change, will switch to a different reinforcement schedule. Okay, so you can predict, you can tell the animal what schedule it's on by giving it a discriminative stimulus, S prime D here. Discriminative stimulus signals what reinforcement schedule or what reinforcer would be available. So if you present a green light in a chamber, the animal you can teach that animal to associate with an FR1, but then it switches to a white light from a green light, and then all of a sudden it changes the context. That's a discriminative stimulus, the light changing. Discriminates one context from another to tell the animal that there's a different schedule, a reinforcement. S delta, or the Greek sign, triangle, the delta, the stimulus, essentially a discriminative stimulus like a different colored light that will signal no reinforcement happens. So you can get animals to simply do an FR1 and get that behavior that I showed you in the other graph, continuous behavior under a green light condition. You switch it to white light, the animal will just stop responding because it's learned that there's not going to be a reinforcement in that context. Okay? So you can really study environmental effects on reinforcement with these sorts of schedules. Oh, I should put this up front, but this is essentially the definition of multiple schedules. 2 or more schedules that alternate and are signaled by different discriminative stimuli. It could be tones, sometimes the discriminative stimulus is the way the animal feels, the internal feeling of the animal about the drug you give. So for example, there's something in behavioral pharmacology called drug discrimination. So under drug discrimination, you train the animal, it receives reward only when it feels like, if you want to understand how closely some compound that you make is aligned to how nicotine feels, for example. You give the animal nicotine, and then you train it to get reward on some lever, right, that it likes, versus another lever that doesn't have reward, right? And then you train it over time with different compounds that it only is going to be reinforced by, when you give it saline, it gets reward on, it doesn't receive reward there. So then it learns and associates the internal feeling of nicotine with getting reward in a particular area. So if you give another drug that is not quite nicotine, you can ask whether it feels like nicotine to the animal, because if the animal goes and tries to engage that behavior in that discriminative context, you know the animal thinks it's nicotine even though it's not. But then you can give a drug that's, and this is where you start getting drug classes. This is how you got drug classes and how we understand it. You have hallucinogens, psychostimulants, you have opiates, and all these sorts of things. That was often done by all those sort of things sort of lump in a drug discrimination. Essentially, not only self-reported humans, but animals have told us that they feel very consistent There's some important principles of, this goes till 10. Okay. So important principles of behavioral pharmacology, rate dependency. You don't have to know this for the test. If anybody wants to talk about this, I'm happy to talk about it, but I'm sort of going to fly through some of this. Behavioral effects of the drug are not always consistent from one individual to another, okay? It's not like amphetamine does this to everybody all the time, right? They're very good. And sometimes the behavioral effects of the drug, behavioral pharmacology, depend on a lot of different things. One of them is rate dependency. So if you have an animal pressing a lever for sucrose or some sugar pellet, let's say, and you want to see how amphetamine changes that responding for that reward. Behavioral effects depend on the ongoing rate of behavior. So some animals will press a lot, for example, and have high responding for something like sucrose, and other animals will have very low responding. The same dose of amphetamine could either increase or decrease rate of responding depending on how the baseline. Okay, that's rate dependency principle. And this is when responding is similar. In other words, there's no rate dependency. Everybody's responding to like sucrose the same. But however, the behavioral effects can vary depending on what it is that you give them in terms of a reinforcement. Do you give them a sugar pellet? Do you give them, are they trying to avoid a shock? Those are two very different reinforcements. The effects of drugs, environmental context, depending on environmental variables. I've already talked about discrimination. You know, a green light versus white light coming up on the in the chamber can change how animals will behave, and then the behavioral history. So not only the biology, but essentially the drugs in which the animal received in the past, and what behavior that animal has done in the past. So really quickly, rate-dependent effects. A lot of this was shown in the original. This is B.F. Skinner's work, who basically came up with a lot of these theories, and was sort of the grandfather of all this, the pioneer. Subject for Harvard Pigeon Labs. Okay, this was the chamber that they initially worked under. Looks a lot like a med-social chamber now. This was the computers that they used. You can see all these wonderful. This probably fits in your phone right now. This entire room. But essentially, what they have is these cumulative response graphs that are essentially drawn out by essentially a pen, sort of like a earthquake or Richter scale type thing, where it's like looking at seismic activity. This is looking at response rates of pigeons through essentially these computers here. So the pigeon would peck a key for some amount. You have a switching circuit, then you have an ink writer writing the different events that are happening, and then food would be delivered as a reinforcer. So basically, so this is a 15-second fixed interval schedule versus a fixed ratio 50 schedule. You get a lot more responding on the, we've already talked about this, a lot more responding in pigeons 1 through 4 on the fixed ratio. Okay, so a lot more responding here as a mean relative to this, or just normal. Then let's see, they gave them phenobarbital, okay, and so they showed no effect on this initial responding. No effect observed. So this is what I was talking about. Behavioral pharmacology has to be more quantitative. There's no qualitative visual difference. So the dose of phenobarbital intramuscular 1 mg, no effect on any of the birds. 2, no gross effects, and then 4, you see marking coordination. So it really has an effect. You need to separate a lot of your behavior from gross disturbances in behavior pharmacology. But look at the doses 1 and 2 that had no observable effect in by the experimenter. You see that under a fixed ratio 50 relative to baseline responding, phenobarbital actually increases responding, okay, for the food in pigeons under a fixed ratio 50 schedule. But under a fixed interval schedule, it actually significantly decreases responding, okay? So the schedule of the reinforcement which the animal is on will determine what effect the drug has, okay? So schedules itself can determine whether you have increases in behavior or decreases. And this is under a condition in which there's no observable effect from the experiment. This is another way to plot this. This is sort of amphetamine. Okay, so this is the control rate of responding before a drug was given. Okay, so you have very low rates of responding. When you give an amphetamine subsequently on that, let's say food responding, it goes up to like 1,000% of baseline under low responding conditions. But with amphetamine, if you have very high rates of responding at baseline, way up here at 3, then amphetamine will actually drop you to 50 to 25% of baseline. Okay, so it depends on the. So it's a very nice linear relationship between the initial rate of So that's rate dependency, okay? That's just the principle of rate dependency. Maintaining event, depends on the types of reinforcers. So initially in earlier studies, the rate, like I just said, the pattern responding seemed more important, but over time, they manipulated other parameters, okay, and showed that the type of reinforcer also changes the effect. So for example, this is your doses of D-amphetamine that were given to an animal and the amount in which it would respond, okay? In this case, in the closed triangles, it's responding for food, and amphetamine increases food responding to some degree up until you get to high doses, okay? And then, interestingly, if you can see that if the animals are trying to, if you have shock as a reinforcer and the animals are trying to avoid shock, you can get their response rate to avoidance is a slightly different curve here of the increase in avoidance, and then it drops down to be negative, right? Oh, sorry, this is fixed interval. Then if you look at the fixed ratio, you see no increases in fixed ratio and actually a decrease of the high responses. So fixed intervals of shock and food show increases in behavior. Fixed intervals show decreases in behavior. I'm going to go over that for the sake of time. The drug itself, or the reinforcer itself, the drug itself can change it. So for example, food and shock avoidance, D-amphetamine increases shock avoidance in food, rate of responding across dose response curve, but morphine, a different class of drug, will increase shock avoidance but decrease food maintained responding. So it depends on what drug and what drug we give. Same thing here. Food, again, shock. Certain drugs, alcohol, CDAP, cocaine, phenobarbital, all increase food maintained behavior but decrease shock avoidance behavior, shock maintained behavior. Except for cocaine, increases both. Let's skip that. So basically, Barrett summarized a lot of this work with the type of reinforcement we get. It didn't contradict earlier finds on rate dependency, but they it's several more questions now. It gets a little more complicated about the predominant view, where they were saying that the effects were largely independent of the type of reinforcement and only based on the animal's pre-existing rate of response. So essentially, fixed interval schedules were different than fixed ratio schedules. Okay, so it depends on the type of reinforcer that's allowed. I'm not going to go over that. Just more of the same thing. Running out of time here. So the environmental context. Let's talk about that, and the behavioral history of the animal can change reinforcement. Let's talk about that a little bit. There's several types of principles associated with environmental context. Context, there can be interactions between contexts. So change in behavior during the presentation of one stimulus, it can be brought about by changing the schedule with a different stimulus. Okay, so these two things, so basically two different schedules of reinforcement across a multiple schedule can influence each other and as opposed to having them independently. Okay, that's sort of interacting. Behavioral contrast in terms of those interactions is when the rate of responding during one stimulus is a direction away from the rate of responding during the presentation of other stimulus. So two stimuli can diverge in how much rate of responding they will get even within the same animal, or response induction when the two rates of responding go in concert with each other, when ordinarily independently they may not. So this is the change in context. So how, similar to what I was just saying about multiple schedules, how does fixed ratio influence fixed interval and vice versa when given in multiple schedules as opposed to just independently of one another? So they had a blue light, for example, that would signal that the animal can take a fixed interval after 3 minutes and get a reinforcer, whereas the fixed fixed ratio, which could be either 30, 90, or 150 in that change, but it was signaled by the red. Okay, and you get rates of responding and it alternated between the two based on the context or the discriminative stimulus that the animal was in. And what you see is that this is fixed interval response rate, and depending on whether the animal got a fixed ratio of 30 in between each of those fixed intervals, a fixed ratio of 90 or a fixed ratio of 50 changed the way in which the animal would behave under the fixed interval. Okay, so the fixed ratio changes the behavior under a fixed interval. Okay, independent of how the fixed interval would happen in context alone over doses of ethanol. Okay, so that's one example of how context or the environment of one schedule can influence another. This is more context, and this is sort of right before avoidance would happen to a shock. Amphetamine sort of increases rate of responding. But after avoidance, you see much higher shifts in rate of responding after the animal has successfully avoided shock. This is just more examples of where the animal is in the neurobiology physics at a particular time can change the effects of drugs. So this experiment and other indicate the reinforcing or punishing properties of environmental events are not invariant features of the event, but depend also on characteristics of the schedule under which the event is presented. Okay, other factors such as the behavioral history of the organism and environmental context in which the behavior occurs also play a role in determining effects and various consequences on behavior. Another concept in behavioral pharmacology is the idea of behavioral tolerance. So tolerance to the effects of drug occur because of interaction between the drug effects and the consequences of ongoing behavior. Tolerance meaning less effectiveness of drug in a particular objective measure or outcome measure. So here's an example of tolerance here. We'll skip to it. So basically, under control, what animals will do, this is one of the histograms, so this is responses. This bar from here to here, this would indicate a scale of 1,000 responses. So D-amphetamine, under control conditions, the animal will essentially pack a lever, right, to receive reinforcers. The first injection it's ever seen, you'll get increased rates of responding. The second injection of the same dose, increased responding, but not as much, and you get tolerance over time. So by the fourth injection, the animal looks almost like the control after 4 injections of amphetamine. So you have tolerance, okay, to the rate increasing effects of amphetamine. Another example of this is sort of fixed interval. Animals were trained under fixed interval set schedule and looking at an amphetamine dose response curve from 0.25 to 1 mg per kg or a different schedule. Okay, depending on the schedule it was, some animals almost showed a resistance to tolerance and other animals showed, depending on the initial schedule, great tolerance. There's specific tolerance over chronic 7-day amphetamine treatment to IRT schedules relative to FI schedules. So it just depends sometimes on the schedule. Now, very briefly, in the last, probably let you about 15 minutes early, but in the last 15 minutes, I want to talk about models outside of what a lot of drug addiction work uses. And there's several types of models, a whole host of models really used in the literature even today. And they can sort of be grouped into 3 different types of models. Predictive. So predictive models do not resemble the disease at all in terms of etiology or symptomatology. Okay, but it is predictive of clinical outcome for reasons that may or may not be known. Okay, I'll give you examples of predictive, and most I would say are predictive. Okay. Isomorphic resembles the disease in terms of symptoms and predictive outcome, but is artificially produced in the lab. Isomorphic probably falls along the lines of more of the drug self-administration that you probably learned about yesterday and today. So in other words, we'll let animals press a lever to receive cocaine, and it will engender rates of responding similar to humans. So in some ways, in terms of the symptomatology, it sort of mimics what happens in humans, but it's artificially produced in the lab. Then homologous, which is essentially resembles the disease in terms of etiology, symptomatology, and predictive outcome. This is incredibly difficult, if not nearly impossible, in animal models to have homologous models of humans. But I guess it happens in very limited cases. So models of affective disorders. So I'm specifically going to talk about behavioral models that you might employ in your research for depression, anxiety, and schizophrenia. We've already talked about models of abuse, liability, or addiction. I talked about condition, place, preference. I think I briefly brought up drug discrimination, and of course self-administration under all those schedules of reinforcement. So we're not going to go over these, but we've already talked about them. I'll talk about that last. So animal models, some animal models of depression. Now this is the isomorphic that I was talking about, whereby there's no relationship to the etiology or symptomatology of depression. But for whatever reason, antidepressants that work in these models work well to be an antidepressant in humans, okay? Does that make sense? So the forced swim test, or tail suspension test in animals, for example. So antidepressants have amazing efficacy in like something called a forced swim test, where you put an animal in like a little pool for it to swim, like a rat, and see how long it will swim and try to get out, and then versus how long it will float in that time. And so the degree to which a drug will increase the amount the animal will swim rather than float will actually be very predictive of how well it will be used as an antidepressant. Okay, there's nothing to do with swimming and depression, right? But there might be some underlying neural circuits that overlap with each other. Now people retroactively go back and attribute sort of an anthropomorphize the rat in some degree. Oh, the rat is learned helplessness, so it floats. And so you're avoiding learned helplessness by making it swim more, giving it more hope. You know, these sorts of things don't work really in animals. The idea of modeling hope is, but people like to put these things on animals. It's not really tested in the lab. Really, outside of neural mechanisms, nobody really knows why more swimming leads to clinical efficacy and less depression. Learned helplessness, I sort of talked about that. There's other models of that I won't go into. Other models of depression, you can sort of induce a depressive-like phenotype that's more, not so isomorphic, but actually can exhibit some examples of stress, so lack of grooming, reduced sucrose intake and things like that while giving chronic mild stress or early life stress to animals. Some people do that. DRL2, so this is that DRL schedule where the animal is not allowed to respond until a certain amount of time in order to receive a reinforcer. The amount to which a drug will increase the ability of the animal to do that can actually predict antidepressant efficacy. So the better the animal becomes at withholding its responses for 72 seconds before it receives a reinforcer will actually predict efficacy of drugs. Others, there's withdrawal-induced anhedonia. So withdrawal from opiates creates an anhedonic state that mimics depression, for example. You can do lesions of the olfactory bulb, and then you have breeding and genetic approaches that will lead to a depressive-like phenotype. So this is like the DRL 72 that I showed you. Clinically effective antidepressants decrease response rates. Okay, so the animal's rewarded for decreasing response rates. It's supposed to wait 72 seconds and not respond at all. Every time it responds within 72 seconds, it's penalized by restarting the clock over again. So it decreases response. So dicipramine, which is a clinically efficacious antidepressant, will decrease response rates and then and by definition increase the amount of reinforcers it will receive over time. So dicipramine will essentially both act as an antidepressant in humans and actually make animals better, the DRL 72. This is an anxiolytics can be predicted, for example, by very mild but somewhat aversive. Still, the animal doesn't want to receive a shock, so the animal receives food pellets under these reinforcers. You can see that the animal's responding is going up, up, up. It'll receive a reinforcer, then it'll end, and then you'll pair the food pellet that it once received with a shock. The animal will essentially terminate responding because it doesn't want the mild shock. Okay, buspirone, which is an anti-anxiety, will then predict the animal will continue to respond despite being punished, and this predicts efficacy of anxiolytics. And then you can do, in the same task, for example, you can do comparison of one drug with the other and look at the potency. So here, buspirone is much more potent than diazepam, right? So this might have a better indication for anti-anxiety even before you test it in humans, and lo and behold, it ultimately does, right? So there's another thing that you come up with, diazepam, which is even better than buspirone. Buspirone, you do that in animals first, and you don't have to test it in humans, and lo and behold, it's more efficacious and anxiolytic than it is buspirone from this one test, and it really has nothing to do with anxiety necessarily, anti-anxiety. Schizophrenia models, so there's both positive and negative symptoms of schizophrenia. It's a very complicated disease. I'm not going to talk too much about it. You probably already talked about it in some context. It's a high incidence in the population. Suicidal ideation, typical onset in late adolescence. Positive symptoms include meaning the existence of something that isn't normally there, such as hallucinations, delusions, formal thought disorder, bizarre behavior, disordered thinking, disorganized repetitive behavior, and then negative symptoms, meaning the lack of something that should be there in a healthy population, flattened affect, anhedonia, or essentially a lack of interest or a lack of reinforcement and rewarding potential impairments. Classic one from the 1950s, condition escape avoidance, again a mild shock, and then the rat can jump up on a pole to avoid the mild shock. So So this was done even in the 1950s. So this essentially these types of studies help people get out of the asylums I was referring to before. These were sort of the initial studies that allowed this show antipsychotic efficacy. So they did a study where independently a clinician ranked behavioral profile of all these neuroleptics or antipsychotics by how well it worked in humans, 1 to 7, and then independently of that, we looked at the conditioned avoidance response that I just showed you. So the ability of the animal to avoid more, the efficacy of that particular behavior across these, and it lined up perfectly. So even though this has nothing to do with schizophrenia per se, it can predict the ability of these drugs to work in schizophrenia. I'm not going to go into negative symptom. So there's basically antidote, such as reduced sucrose consumption that you can do in animals, and so to withdrawal, things like that. And I want to end it on this, I meant to put this last. And so these are just very briefly, in the last 2 minutes, the problems that we're facing with behavioral pharmacology at this point in time. One problem is, of course, essentially what happens initially is you can have for every 10,000 compounds that one is screened in the lab, right, during the drug discovery stage 1 phase. Through these models, you start to narrow it down, right? And then from 10,000 compounds just in the preclinical phase, you can already get down to about 250 compounds to test more sophisticated the models that we're looking at here, right? From drug screening, medicinal chemists develop the drugs, you put it in behavioral animal models before you put it in humans. And then you have what's called an investigational new drug that can be submitted, okay, to go into clinical trial because you've done all the tox screening, you've done all the behavioral measures that we're talking about here. It can be predictive of some sort of efficacy later on. And you're already down from 10,000 compounds to, down to 5, okay, just from the preclinical stage. So this is a huge filter, okay. For every 10,000, you have like 5 compounds. And you have a phase 1 where it goes into humans, where you have 20 to 100 volunteers, okay. It's narrowed down even further. Then you go into a phase, you pass the phase 1 clinical trials, you go into phase 2 clinical trials, where you can bump up the number of volunteers from 100 to 500, and then phase 3, where you go from 1,000 to 5,000. You get down, and ultimately you can get, if you're lucky to a new drug discovered that's FDA approved, number 1. So for every 10,000 compounds you may have screened out, you likely, if you want to invest the billions of dollars that it takes to get from here to here, you can likely get to 1 new drug, okay? So this is where all the models that we're talking about today are essentially here. So there's no way there's money available to screen 10,000 compounds in humans. It's not only a safety issue, it's about the fact that it just won't happen, essentially, without these early models. One problem, though, behavioral pharmacology is having right now, especially the preclinical models, many of the currently marketed drugs are direct results of what I was talking about in the 1950s, okay? Prozac, Clozaril, Effexor, all these are based off of stuff that was come out in the 1950s, very small modifications. So the initial drugs served as the basis for development of animal models, biochemical assay systems, so everything was built around the initial efficacy of these drugs, which is sort of a side effect of being problematic when you want to come up with 3rd and 4th generation drugs, right? So how do we get so Forced Swim test predicts antidepressants really well, but antidepressants only work in 50% of the population. How do we get the other 50% of the population when we've maxed out our ability to test with Forced Swim test? We have no other models to necessarily move us from 2nd to 3rd generation, right? We need other behavioral models to test the efficacy. So there's a lot of approaches, such as individual differences in these preclinical models that people are looking into, things like that. But a lot of these, the dopamine hypothesis of schizophrenia and addiction, the biogenic amine hypothesis of depression, the GABA hypothesis of anxiety, all came out of these initial studies from the 1950s. They didn't even know what a receptor was. They knew that dopamine was involved, and the idea was there was a lock and key mechanism, and then Bob Lefkowitz discovered that in the 1970s and 1980s later on. But there was a dopamine hypothesis of schizophrenia well before that, because drugs that they knew hit the dopamine system, for example, were efficacious behaviorally, right? So a lot of the brain mechanisms that exist came out of the initial behavioral pharmacology that existed at the time and was rooted around that. So essentially we need to have better models to be able to predict the subtle differences now that we're getting into ultimately, like depression, things like that as well, the other complicated factors, it can manifest itself. The clinical manifestations and someone being, from the their own behavior being labeled as depression can manifest from multiple different brain pathways or combinations. There's not a depression circuit in the brain. That's all I have. Is there any other questions? I'm happy to sit around. We have 15 minutes left or so. I know what clock you look at. Yes.
For the conditioned aversive test, for the response one with the shock that the rat is going on like the pole.
Oh, yeah.
So what made them like initially just want to test antipsychotics for that test? What exactly is the behavioral symptom that, like the aversive response, what exactly like is that? I guess clinically.
I actually don't know to answer your question. My guess is it has something to do with the idea. So there wasn't much pharmacology available at that time, first of all, so, you potentially have hundreds of thousands, if not millions of compounds to choose from. You have to be really, you have to have a nice rationale. Back in the day when you have thorazine and chlorpromazine, you might be able to test both. You know what I mean?
Mhm.
So some of it might just become, it was a much simpler world and the availability was there. I'm not saying that they didn't have a rationale at all, that could have easily played into it. But I don't know, ultimately. It's a good question, and it's probably out there. It's just something I don't know. I might look it
Did
up.
they
It's a
test
good
other
question. I don't know.
drugs in that test, or in that model, or just
Oh, yeah,
antipsychotics?
Yeah. And that might ultimately have what it had been. It's not like, I'm just not showing you the negative results, associated with other things. It may have just been, I'm sure probably just about every early drug was used in this. And either wasn't efficacious or, if it was, it predicted its ability to reduce positive symptoms in schizophrenics, for example. So at the time, it was just sort of like the Wild West. Now, it wasn't, it's hard not to think in terms of what we've been told to think in terms of, but they didn't have drug classes and things like that. They may put the drugs on that was only known in relation to the behavioral profile that it would elicit. They didn't really have a concept of the mesolimbic dopamine system, the 1950s, early 1950s, right? And back in those days, there were, very crude, like even lesion studies to understand that, right? Going on to parallel, right? Just in humans too, unethically. And a lot of lobotomies, just see what happens, But interestingly, a lot of, there's TMS going on, transmagnetic stimulation. Colleen Hanlon, who was a postdoc here in Linda Perino's lab, is now becoming famous for that. In a lot of ways, it's not an aversive. You wouldn't even notice it. I've had friends who do it, but it basically, for milliseconds, deactivates and scrambles an entire hemisphere of the brain. And they're taking it back to akin to like the electroconvulsive therapy that they used to use in asylums back in the day that was so efficacious for depression. It really did work. People who were depressed in these asylums and could not function would actually be revived, but it would have to be, it didn't have much efficacy in terms of how long it would last. So they would have to stay there and receive it. And now, for it's being looked at again for the transmagnetic stimulation, where you just take a magnet and scramble the brain for milliseconds. And you don't even notice anything. It can change craving, and it can bring antidepressant symptoms back up and things like that. So there's a lot of things from the 1950s they were doing that, now we're more sophisticated in the pharmacology and we're more sophisticated in the technology. But this is all much more translational than, I was at a journal club yesterday that was talking about how kappa opioid receptors signal glutamate terminals from the ventral hippocampus different than the BLA, and how that ultimately influences D1 versus D2. That's wonderful work and it's in nature, but what does that mean in terms of translation? I mean, I'm not, these people are funded by the National Institute of Health, and their mission is to increase health in humans by definition. So I'm not saying we shouldn't understand the brain, but the higher impact work now is really getting into things that may not ever be able to be touched in terms of actual health is concerned. Or hopefully it will set a precedent to increase the technology to be able to go at these very specific things. How do you dissociate a kappa receptor on one terminal versus a kappa opioid receptor on a different terminal in the very same brain region? With pharmacology, it's likely not going to be a pharmacological approach. Any other questions?
I'll stick around for 10 more minutes if anybody